This conference will now be recorded.
Good evening, students. I'm Dr. Deepa from TeenEinstein. So today we are going to start our next unit. Okay, next unit in the sense we are moving to next topic. Biotechnology. We are moving to a different topic from this, biotechnology. In this we have two chapters. First one is biotechnology, the principles and the processes. What are all principles behind this biotechnological processes and how we are applying that principles for different kind of processes, or what are the processes and principles included in this biotechnology. The next chapter deals with biotechnology and its application. Okay, next the application level of all these theories and concepts and all. That's what we are going to learn in the second chapter. The first chapter, biotechnology, principles and processes. We are starting today a new concept. So chapter 11, biotechnology, principles and processes, part 1, we are going to start now. See, this chapter is very important for your NEET exam because you'll get so many questions. This is the new concept for you people. So you'll get different kinds of enzymes name and the technologies which are using. So for all these things, it's new to you people, so you have to be careful, especially for NEET exam. So let's start. What is biotechnology? Like from that word, from this itself, biotechnology, biology and technology. Okay, combination of biology and technology. How this biology and technology is working? Biology plus technology. Okay, it is the integration of natural sciences and engineering sciences in order to achieve the application of organisms, cells, parts thereof and molecular analog for products and services. Okay, that means we are combining the natural science and the engineering science. The engineering, the technology we are applying with the help of this natural science theories. So what are all applicable for natural science, that theories, we are with the help of that engineering science, we are going to make a new, that means the application, how we are applying. See, there is just like biological theories we are reading, how to apply that in day-to-day life and how to make a new item or whatever we can use, whichever is useful for human beings. So that's how we are making use of these technologies and biology and all. Okay, so biotechnology, the use of biology to solve problems and make useful products. The products, we need to make useful products by applying this biology and engineering science. Okay, how to apply this biological theories with the help of engineering science and how to make useful products. That's what we are going to learn from biotechnology. So nowadays it's very important. It's a prominent area, the biotechnology, and from that the major products, therapeutics, therapeutic proteins and drugs with the help of genetic engineering. Okay, so with the help of genetic engineering, so mainly now you know that vaccine production, other drugs, everything that is all coming under biotechnology. Okay, then different applications in agriculture sciences also we can see for high production and for disease resistance. All those applications all are coming under biotechnology. Okay. So many processes and techniques that are coming under this biotechnology. So many are included in this biotechnology, like genetically modified organisms. So, which are now, for example, we can say genetically modified organism. Okay. Then in vitro fertilization. In vitro fertilization, that for test tube baby and all, in vitro fertilization. Then synthesizing a gene and using it. Synthesis of new gene, how to synthesize a new gene. New gene means each character is controlled by a gene, right? So how to synthesize a gene which we need a desired character, right? So for that, how to synthesize a new gene to control that character, or how to synthesize a new gene and how to make use of that character which is controlled by that gene, right? Then developing a DNA vaccine or correcting a defective gene. DNA vaccine development. Now we know that the vaccine development, importance of this vaccine development. When this corona, this COVID, all these vaccine development for different kind of diseases. So how to develop a vaccine? So that is the importance of such things nowadays we know very well, right? Due to this corona, COVID and all, we can understand how important it is. Okay. Then correcting a defective gene. A defective gene that may cause different kind of diseases. A gene, a problem occurring to a gene in a body, that in a body, it may be a plant or animal, whatever may be, that can create a lot of issues, right? Everything is related. In organism, everything is related. So one defective gene that can cause several diseases or several problems in an organism. So what, correcting such defective genes, all these are coming under biotechnology. So biotechnology is very important. And you know very well, nowadays food, it is very less, but the population is more. So we can't increase the land area. So what we need to do, we need more production. From the same land, we need more production. So earlier and all, what people were doing, according to the season, they were sowing, they were harvesting. But now, is it possible? Can we wait up to that? No, it's not possible, because we need a large production to serve this population, right? Because population is more now. Every day it's increasing, day by day it is increasing, the population, and the food production also very important, because that should meet the demand of that food, right? So what we need to do for that? We need to increase the production. So we need higher yielding varieties, disease resistant varieties. Then another one, an important thing is climate. So according to climate, we can't wait. So we need, for every season which is giving the maximum yield, such kind of crops also we need to meet the food demand. So what we need to do, we need to change the characters of crops, and we need to, and everything is very important. And the major thing is nowadays water is very less. So drought tolerance, that character we should bring to each crop. Drought tolerance, salinity tolerance, these are all the major research areas. Okay, so such things and all, nowadays it is demanding, because food is very less and the population is more. So to meet the demand of food, we need to make more food, or the land area that I said, it's not increasing. So to meet the demand of population for food, we need to create crops which are higher yielding and which can tolerate drought, which can tolerate salinity, and which can give more yield. Okay, so all these are, with the help of this biotechnology, it is going on. All this research research are going on. So it is very, very important. Biotechnology is having more importance in nowadays life. Okay. So genetically modified organism to achieve same on a larger scale. This is what I said, we need more. Okay. And in vitro fertilization leading to a test tube baby. For test tube baby, in vitro fertilization, there comes the demand of that. Then synthesizing a gene and using it for a special character, synthesizing a gene and how to use it. Then developing a DNA vaccine. Nowadays vaccination, it is very important. So developing a vaccine, how to develop a vaccine, then correcting a defective gene. All these are done with the help of biotechnology, biology and technology, both combination. When it comes, it makes useful products. Then next is European Federation of Biotechnology that has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. Okay. The integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services. That is the definition of biotechnology according to European Federation of Biotechnology, EFB. Okay, European Federation of Biotechnology. So this explains the modern molecular biotechnology and the traditional view of biotechnology. The integration of natural science and organism. Okay. So for what and all we can use this biotechnology to improve the yield and disease resistance, then for fortified crops, then reproduction in aquaculture, pest resistant crops, biofuel, enzyme, fermentation for improved yield, all these. So here you can see different applications of biotechnology for improved yield, fermentation, enzymes, biofuel, pest resistant crops, then reproduction in aquaculture, fortification of crops, then disease-free plants. Everywhere you can see the application of biotechnology. So as an introduction, we can say that much about biotechnology. Then we are moving to principles of biotechnology. Okay. So what are all the principles of this biotechnology? So mainly there are two techniques. Okay, two techniques that show the modern biotechnology, the growth of modern biotechnology. One is genetic engineering. The first one is the genetic engineering. Main two core techniques. First one is genetic engineering and second one is the bioprocess engineering. Genetic engineering and bioprocess engineering. Genetic engineering, that means the engineering in genetic material. That means changing the genetic material, making alteration in the genetic material. Bioprocess engineering means for the growth of that microbes, then for the production of this antibiotics, enzymes and all in large scale. Okay, that is bioprocess. Making use of different biological process in large scale. Okay, that is called as bioprocess engineering. Genetic engineering means making alteration, engineering done in the genetic material. Okay, so genetic engineering and bioprocess engineering, these are the two main concepts or main techniques which explains the biotechnology or modern biotechnology. That is the modern techniques which are used in biotechnology. So first one is genetic engineering, second one is bioprocess engineering. So genetic engineering, let's see genetic engineering. That is What is genetic engineering, or what we are doing in genetic engineering? To alter the chemistry of the genetic material, either it is DNA or RNA, whatever may be. See, DNA organisms are having DNA as a genetic material, and some are having RNA as a genetic material. Okay, so these techniques, genetic engineering techniques, for what purpose we are using? To alter the chemistry of the genetic material, either it is RNA or DNA, whatever it may be. We are changing the genetic material chemistry, the chemistry of that genetic material itself changing. For what? To introduce those into host organism and thus change the phenotype of the host organism. Then after change, see, normally if we have a DNA or RNA, it will be having specific genes and it will be having specific characters. So if we want some kind of alteration or changes in that character, with the help of genetic engineering, we are making changes in the chemistry or making alteration in that genetic material. In DNA or RNA, whatever genetic material it may be, we are changing that genetic material, chemistry of that genetic material, and we are making changes. We are introducing that new changes into that host organism. So that host organism, according to the newly changed genetic material, it will show the phenotype, or according to the DNA or the genetic material, all organisms are showing the characters, right? It depends upon the genetic material, the character. See, if you have dark hair, the specific gene will be present in your DNA, right? If you have white hair or golden hair or eye color, everything matters. Everything is regulated by gene, right? So that genetic material we are changing and again introducing that into that host organism. So the phenotype or the expression of that gene will change, right? So the host organism's character will be changed. So the genetic engineering, with the help of this genetic engineering, what we will do, we are altering the chemistry of the genetic material. The genetic material itself will get changed, or the chemistry of the genetic material is getting changed, and again introducing that into host organism, but that time the host organism will show the changed expression, or the phenotype of that host organism will change. Okay, here then see the DNA genetic material, some changes are made in that genetic material. So next expression, next time when these genes are getting expressed, the expression will be different. So whichever we need, whichever character we are expecting, according to that we can make changes and introduce into that organism so that it will get expressed in that way. Okay. Next is bioprocess engineering. Bioprocess engineering. Bioprocess engineering means maintenance of sterile ambience in chemical engineering processes to enable growth of only one desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes and all. Okay. So bioprocess engineering means one type of organism or one type of microbe, whichever we want. Okay, so growth of only that one in large scale for some kind of enzyme, some antibiotics and some vaccines without any contamination. That means we are assuring the growth of only one type of organism which is producing some kind of antibiotics or some kind of enzymes which we want in large scale. We are going to produce that. How it will be? Contamination free. Contamination means there is no other type of organisms included in it. There is no contamination. Only the single type of organism or single type of product will be formed only from one type of organism which is grown in large scale. That will be maintained. Large quantities. For what? For what purpose we are going this large quantities of such kind of organisms or microbes? For manufacturing of these products like antibiotics, vaccines, enzymes and all we need in large scale, right? For large scale product we need more organism which is producing that particular enzyme and all. So what we will do, we'll grow that in large scale, but we should make sure that there is no other product or there is no other organism in that, or that is not contaminated with any other organism. Okay, so we are maintaining the sterile condition in chemical engineering processes, or we are maintaining it as contamination free, and we are enabling or we are assuring the growth of only one desired microbe or one whichever particular type organism we want. We are assuring the growth of only that kind of eukaryotic cell in large quantities. Okay, in large quantity production, that is the main thing. In bioprocess engineering, we can produce in large scale, single type of organism, whichever we want. And that helps in the production of antibiotics, vaccines, enzymes and all in large scale. Okay, so there are two main techniques in modern biotechnology, that is genetic engineering and bioprocess engineering. Then, the conceptual development of the principles of genetic engineering. So, the technique. So, what we can understand or what we know about the conceptual development in the principles of genetic engineering. See, we know that the difference between asexual reproduction and sexual reproduction, right? So in asexual reproduction, in sexual reproduction, let us compare the sexual reproduction and asexual reproduction. So in sexual reproduction, there is a male parent and female parent. So there is a combination, right? So there comes slight changes. There is a chance of variation or chance of changes because from different parents, half of the chromosomes, it comes from half from the male parent and half from the female parent. So there is a chance. Asexual and sexual, right? So in sexual reproduction, 50 plus 50, 50 from male parent and 50 from female parent, right? So this is a combination of characters, right? That comes the 100%. So when it is coming to 100%, there is a chance from one parent, from mother, one child it is forming means then half of the contribution is from father, that means male parent, right? So there is a chance of slight variations, different forms of characters you will get. So genetic different combinations you will get, unique combinations, genetic combinations you will get, and in that combination you can see some may be helpful, that means some may be beneficial to that organism, but some may not be useful. So like that characters are there. Some will be plus or beneficial to that whole organism and population. But some may not be useful. Like the different types of combination you will get. But in the asexual reproduction, what you can say, in sexual reproduction, you will get different combinations and useful combinations also will be there, and without that also you can see, right? But there is a chance of different combinations. In asexual reproduction, what you can see, asexual, however the mother is, like that, like the parental organism, exactly, the offspring will be exactly similar to the parental organism. Means whatever character is there in the parent, that will be preserved. There won't be any changes. Exactly the genetic information will go to the next generation also. There won't be any changes due to some other reasons like slight errors and all small variations will be there. Other than that, there won't be any change in the asexual reproduction. But in sexual reproduction, there is a chance of changes. That means various combinations you will get. So asexual reproduction preserves the genetic information, right? This preserves the genetic information, asexual reproduction. But sexual reproduction, that allows variation in different combination you can see. So you know that hybridization, traditional hybridization which we are using in the multiplication of what in animal breeding we are using, and plant breeding also we are using traditional hybridization. So that includes what and all, like along with the desirable genes, undesirable genes also will multiply, right? That means we can't remove all these things. We can make different combinations. See, in one male parent, one character is there. In another female parent, another character is there. So we need the both characters in next generation. Means we can combine. We can join that characters to sexual reproduction. But there, another problem is also there. Through this traditional hybridization procedure, what will happen when we are making the combination in the next generation? There also, along with these desirable genes, some undesirable genes also may pass from the parents to offspring, right? See, there is no use with some characters, but that also will pass to the progeny along with the useful characters in sexual reproduction. In asexual reproduction, there is no changes exactly. The genetic information is passing from parent to offspring. But in the case of sexual reproduction, in traditional hybridization, what is happening? Along with the useful genes, other genes which are not at all useful, that is also passing to next generation. Okay, along with the desirable genes, undesirable genes are also passing. But here comes the importance of genetic engineering. Through genetic engineering, what you can do, when these in sexual reproduction we said desirable and undesirable, both genes are passing to next generation. With the help of this genetic engineering techniques, we can create a desirable part. Okay, so the techniques of genetic engineering, that means the creation of recombinant DNA. Okay, recombinant DNA, recombination. We are recombining. From the normal combination, we are making a desirable combination. Recombination we are making. Recombination of DNA. Then use of gene cloning and gene transfer. So all these, the gene recombination of gene, recombinant DNA, then gene cloning, then gene transfer. All these techniques, these are helpful for what? To isolate a single character. We need a particular character, so we can isolate that, and that we can introduce only one or a set of desirable genes without introducing the undesirable genes into the target organism. That is the importance of genetic engineering. If you want a particular character which is controlled by a desirable gene, we can isolate only that gene we can isolate, and or along with that we can isolate a set of character or set of desirable genes, and without introducing the undesirable genes we can isolate and transfer only the desirable genes into a target organism. In normal or traditional hybridization, what is happening? Along with the desirable genes, undesirable genes are also passing, because there we can't control, we can't limit the passage of genes. But here in recombinant DNA or in genetic engineering, what we can do with the help of genetic engineering, recombinant DNA we can produce, and gene cloning, then gene transfer. All these technologies are there to help. Okay, recombinant DNA, then gene cloning, gene transfer. So we can select the desired genes and we can transfer only those genes to the desirable to the target organism. Okay, so the target organism will be having only that desired character, and we can avoid the undesirable genes completely. Okay, so that is the difference between genetic engineering and traditional hybridization. So genetic engineering that allows to isolate and to introduce only the desirable set of characters or desirable set of genes without introducing the undesirable genes into a target organism. See, if we are passing a piece of DNA, what we need to do, like if we want to pass a piece of DNA into an organism that is having, suppose it is having a character which we want. Okay, so we are passing that into an organism. There is a, and somehow we are transferring that to a particular organism, X, Y or something. So that piece of DNA, sometimes it may replicate in that organism, right? And mostly that won't multiply in that organism. Okay, sometimes only there is a slight chance of multiplication. But when it is integrated, with that DNA of that organism, that will multiply. Okay. So if we are transferring, roughly we are transferring one piece of DNA into an organism, that piece, there is no chance, like 0.01%. That much only, very less chance of multiplication. Okay. Mostly it won't be able to multiply and that won't be present in the progeny also. But when that piece is integrated with the genome of that particular organism, then that may multiply. Okay, so along with that host gene or host genome, this DNA part also may multiply and that will come to the next generation. Because that is becoming a part of the chromosome of that organism. Then that will be having the capacity or that ability to replicate or transfer into next organism. So in a chromosome, there will be specific sequences or specific DNA sequences. Okay. In a chromosome, specific DNA sequences are there. These are called as origin of replication. Okay, so simply we are passing up DNA sequence, a piece of desired DNA into an organism, it won't multiply. But when it is becoming the part of the genome of that organism, or when it is becoming the part of the genome or genetic material of that particular organism, then it will get the capacity or it is having the ability to multiply. Okay. Only a single piece that can't multiply, when it is becoming the part of that particular organism's genome or genetic material, along with that this also will multiply and that will come to next generation. So in a chromosome there is a specific DNA sequence called the origin of replication, which is responsible for the initiating replication. Okay. So the origin of replication, it is specific DNA sequence which is responsible or which is helping for the initiation of replication. So this is initiating the replication process. Okay. So what is origin of replication? These are one mark question, and for NEET also this is important. DNA sequence, specific DNA sequence which is responsible for the initiation of replication or origin or the starting of the replication. Okay. Origin of replication. Now this is the starting point of the replication. Specific DNA sequence which is helping or which is responsible for the initiation of replication. That is called as origin of replication. So whatever that DNA piece of that or that alien piece which we have transferred to an organism that also need origin of replication. So when it is becoming a part of the chromosome or part of the genetic material, that will be having an origin of replication, right? Otherwise, without an origin of replication, this can't replicate or this can't form or this cannot be transferred into next generation. Okay. So when it is passing to an organism, a piece of DNA or an alien DNA which is passing to an organism, without origin of replication, it can't replicate. So when it is linked with the origin of replication, that alien piece also can replicate and that can multiply along with the host genetic material, along with the host genome. Okay. So this multiplication, such multiplication, that is called as cloning. So while doing that cloning work or cloning, what you can see, that can make multiple identical copies of any template DNA. Multiple identical copies of template DNA. Okay. So an alien DNA which is linked with the origin of replication. When it is linked with origin of replication, what it can do? It can replicate along with that genome or along with the genetic material of that host organism. This alien DNA also can multiply or that can also replicate. So this is called as cloning. Okay. So what is cloning? Along with the genome of that, along with the host genome which is having origin of replication, alien DNA also can multiply and that can produce multiple identical copies of any template DNA. Okay. Multiple copies will be there. So identical, clone means exactly similar. There won't be any changes. So multiple identical copies of any template DNA. Okay. So that is called as cloning. Okay. So this is important, cloning. Then origin of replication. Next. So we are discussing about principles of biotechnology. Okay, the principles behind biotechnology, that's what we are discussing. These are very important for NEET exam mainly. Then, the construction of the first recombinant DNA that emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid of Salmonella typhimurium. Okay. First recombinant DNA. The construction of first recombinant DNA from where it came? It emerged from the possibility of linking a gene encoding antibiotic resistance. A gene which is encoding antibiotic resistance with a native plasmid. Okay. Plasmid of which one? Salmonella typhimurium. Okay. Salmonella typhimurium. Then Stanley Kahn and Herbert Boyer. They accomplished this in 1972 by isolating the antibiotic resistant gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. Okay. So this antibiotic resistant gene, by cutting out a piece of DNA from a plasmid. Okay, what they did, cutting out a piece of DNA from plasmid, and this plasmid which was responsible for the antibiotic resistance. So how did they cut? Is it very easy to cut a DNA? It's not like cutting something, cutting a paper, cutting a cloth. It's not like that. Genetic material, how to cut the genetic material or the DNA? And how to cut the DNA at specific location with the discovery of the molecular scissors. See, molecular scissors, restriction enzymes. These are called as molecular scissors. These are the cutting enzymes. Molecular scissors or restriction enzymes. Restriction enzymes, they are called as cutting enzymes or molecular scissors. So which enzyme is called as molecular scissors? Restriction enzymes. It is called as molecular scissors. With the help of this molecular scissors or restriction enzymes, the cutting of DNA at specific sites or specific locations became possible. Okay. Then this cut piece, so whichever desired part of that DNA that we can cut with the help of this restriction enzyme, then that was then linked with the plasmid DNA. That cut piece of DNA which is having that desired gene, that will be linked with the plasmid DNA afterwards. Then this plasmid DNA is acting as a vector to transfer the piece of DNA attached to it. Okay. So suppose this is the from the genetic material, we need this part which is having a desired gene. Okay, A, we'll mark it as A. So with the help of restriction enzyme, we are cutting this, which is having this part of DNA which is having the desired gene A. Okay, then this will be linked to the plasmid DNA. This is a plasmid DNA, and here this particular genetic material is linked which is having the desired gene A. So this is the plasmid and this is the desired, this is the DNA part which is having the desired gene. Okay. So this plasmid DNA, it is acting as a vector to transfer the piece. What do you mean by vector? Vector we have learned in the case of diseases and all, we have learned about vector, right? Vector means which is helping to transfer, like mosquitoes and all acting as a vector. Mosquitoes, to transfer the malarial parasite, we have learned the role of a vector. That mosquito is acting as a vector to transfer the malarial parasite, right? So like that here, to transfer this genetic material or the cut piece of DNA which is having that desired gene, plasmid is acting as a plasmid DNA that is acting as a vector to transfer the piece of DNA which is attached to here, attached to it. So this plasmid, to that we attach the desired DNA piece which is having the gene A. Okay. So that's how plasmid DNA, it is acting as a vector. Next. So what is the role of the plasmid DNA? Here you can see plasmid can be used as a vector to deliver an alien piece of DNA into host organism. Then this plasmid DNA, which is acting as a vector, that will be inserted into a host organism where that can multiply. Okay. Then, now we are learning about the general procedures, then we will learn one by one. Okay. The linking of antibiotic resistant gene with plasmid vector. The antibiotic resistant gene with the plasmid vector became possible with the enzyme DNA ligase. Okay. Linking of this plasmid vector, that antibiotic resistant gene. See here, just now I said this is plasmid, and here the DNA part which is having the antibiotic resistant gene. Antibiotic resistant gene. Okay. So this is having that, we'll mark it as A, antibiotic resistant gene. For example, we are giving it as A. Now we are marking it as A, that is antibiotic resistant gene. And that particular DNA part which is having the antibiotic resistant gene, that will be linked to the plasmid vector. This is the plasmid vector. This part, remaining part, this is the plasmid vector. So to that we are joining this antibiotic resistant gene, and with the help of which one, we can't stick directly with that plasmid vector. So with the help of the enzyme DNA ligase. Okay. Restriction enzymes are called as molecular scissors. These are glue. Okay. Molecular glue, enzyme, DNA ligase. This is for joining. DNA ligase, it's for joining purpose. There, molecular scissors, restriction enzymes, that is for cutting. Okay. So this DNA ligase that will help to join the ends of this cut DNA, okay, which acts on the cut DNA molecules and joins their ends. So, restriction enzymes, DNA ligase. Ligase is acting as glue, molecular glue. That restriction enzymes are called as molecular scissors, okay. So that makes a new combination. If these are joined, an antibiotic resistant gene and a plasmid vector both are joined, means that makes a new combination, right? So new combination of circular autonomously replicating DNA created in vitro, and it is known as recombinant DNA. There is a recombination, right? A plasmid vector, it is joined with a desirable gene or desirable DNA piece which is having a desirable gene. We are joining with the antibiotic resistant gene with the plasmid vector, and that can replicate, which is having autonomous replicating capacity, that can replicate itself, right? So that is called as recombinant DNA. Okay. So when this DNA is transferred into Escherichia coli, that means E. coli, that is a bacterium closely related to Salmonella that could replicate using new host DNA polymerase enzyme. Polymerase means for polymerization or for making multiple copies. These are the important enzymes which are involved in this recombinant DNA technology. So these 3-4 enzymes you have to learn. Restriction enzyme for cutting. First we are cutting. Then we are doing attaching that with another plasmid vector, okay, which is having the capacity of own replication or which is having the capacity, which is acting as a vector. Then next comes polymerization. We need multiple copies of that, right? For large scale production of this antibiotic resistant gene, we need multiple copies of that. So DNA polymerase enzyme that helps to make multiple copies. Okay. When this recombinant, here the recombinant DNA is formed with the help of desired gene plus that plasmid vector, then recombinant DNA is formed. That DNA, which is transferred to Escherichia coli, E. coli, that is a bacterium, and that can replicate by using new host DNA polymerase enzyme. Okay, when it is getting transferred into E. coli, with the help of DNA polymerase enzyme that can make multiple copies. DNA polymerase enzyme for polymerization. Okay. So the ability to multiply copies of antibiotic resistant gene in E. coli was called cloning of antibiotic resistant gene in E. coli. Okay. So the multiple copies of antibiotic resistant gene produced, or the ability of that E. coli, which is a bacteria, that is having the capacity for producing multiple copies of antibiotic resistant gene. So that is called as cloning of antibiotic resistant gene in E. coli. So here you can learn molecular scissors, molecular glue, then DNA polymerase enzyme, role of all these enzymes. DNA polymerase for making multiple copies, that is for polymerization. Or what do you mean by polymerization? Making multiple copies. Okay. Then DNA ligase, it's for attaching, or it is having a gluing effect. Then molecular scissors, first cutting, then joining, then multiplication. The same gene will get multiplied. Okay. These 3 concepts you need to keep in mind. First you are cutting, whichever gene you want, you are cutting and isolating with the help of molecular scissors or restriction enzymes. Then you are attaching that desired gene with a plasmid vector. So there you need a sticky effect or gluing effect. For that, which enzyme you are using? DNA ligase. That DNA you need to join. So DNA ligase. Then you need multiple copies of that. Right? You need more antibiotic resistant genes. So you need multiple copies of that. Can you hear now?
Yes, ma'am.
Yes, yeah,
Okay.
can.
Okay. So first you are cutting and isolating the desired gene or that antibiotic. Here we are explaining about antibiotic gene, resistant gene, antibiotic resistant gene. So that gene we are cutting and isolating. Then you are attaching that with the plasmid. So you need a gluing effect. So for that what we are using DNA ligase. First for cutting, restriction enzyme, then attaching that with the plasmid vector, that is with the help of DNA ligase, which is called as molecular glue. Okay. Then you are multiplying that. For multiplication effect, you are using another enzyme, DNA polymerase enzyme, that makes multiple copies. Okay. Then that ability to multiply copies of antibiotic resistant gene in E. coli, that was called cloning of antibiotic resistant gene. Cloning of antibiotic resistant gene. Only that here cloning means making multiple copies. Okay, that you know we have already learned about cloning and all, making exact copies, multiple copies. Okay, so here cutting, then joining, then multiplying for large scale production. Okay, so in recombinant DNA technology, these are the main steps which you are following. Then first, the three basic steps which we are using. Okay, which are the three basic steps which you are using for genetically modified organism? So to modify an organism, genetical modification of organism, first we will identify the DNA with desirable gene. Whichever gene we want, that we will find out first. Whichever desirable gene is there, that one we will find out. And the identification of that DNA, the DNA part which is having that desirable gene. See, in this whole genome, we need, see here, this is the genetic material of. In this, we don't want the whole part. Only here, your desired gene which is present. Okay. So we can cut and take only this part. Then, introduction of the identified DNA into the host. We can, with the help of restriction enzyme, we can cut this enzyme and we can join with the plasmid with the help of ligase enzyme. Then we can introduce that into a host. Okay. Then maintenance of that introduced DNA in the host and transfer of the DNA into its progeny. So we are introducing that into a host. There it will multiply. Along with the host DNA, it will multiply. If only this piece we are transferring, it won't multiply. It won't go to next generation because that need a origin of replication, we said. So along with the host DNA, when we are integrating or when we are joining this with the host DNA, that host DNA will be having origin of replication. So with that origin of replication, it will start multiplying. Then it will transfer that host DNA along with the host DNA that will get transferred into next progeny also. So which are the three basic steps? First, identification of DNA with desirable gene, then introduction of the identified DNA into the host, then maintenance of introduced DNA. We need to maintain that introduced DNA in the host and we need to transfer that DNA into progeny. Okay, maintenance of that first, identifying the desirable gene or identifying the DNA which is having the desirable gene, then introducing that into a host. That identified DNA should be introduced into a host, then maintaining that introduced DNA into the host and transferring of that DNA into progeny. Okay, so these are the three main steps that you should remember for making genetically modified organism. Okay. So next, what we need to learn is which are the tools which are using for recombinant DNA technology. So we need to learn a lot about that. We need to learn about restriction enzyme, then polymerase enzyme, ligase, vectors, host organism. So recombinant DNA technology, the main tools which are there in recombinant DNA technology, they are restriction enzyme, polymerase enzyme, ligase enzyme. We have told about the role. Okay, restriction means you can cut the desired part with the help of restriction enzyme. Okay, that restricted part you can cut and take. Then polymerase enzyme for polymerization. Then ligase for joining. Then vector. Vector, you know the role of vector. Then host organism. A suitable host organism should be there. A suitable vector should be there. Ligase enzyme, polymerase enzyme, restriction enzyme. These are the three main enzymes which are used in recombinant DNA technology, and the role of each one you should by heart because it is very, very, very important for your board exam and your NEET exam. Okay, role of each enzyme. Restriction means for cutting desired part, then polymerization, then ligase for joining. Restriction enzyme is called as molecular scissors, and ligase, it is known as molecular glue. Okay. Then in next class, we'll be learning about each one separately. Restriction enzymes, we need to learn in detail. Polymerase enzyme, then ligase, vector, host organism, everything we have to learn in detail. So in next class, we'll be learning everything in detail. Do you have any doubt in this? Is it clear? Is it clear? You want me to repeat any point in this? Okay, hope it's clear. Just go through this. Because if you don't have any idea, you can't go to next topic, you won't understand anything. So just go through all these things, role of each enzyme,
ma'am.
and how these are working. Okay, we will meet in the next class. Thank you.